What I'd like to do today is share with you some interesting findings about the biology of Alzheimer's disease, which is the main disease process causing dementia. I'd like to start with some definitions. What's dementia? What's Alzheimer's disease? What do we mean by those words? And what's different about them and what's the same? How genes in rare families like Amanda's family hold the key to understanding the biology of these conditions. I'd like to share with you some new brain scanning methods that have helped us understand what's happening in the lead up to symptoms, in the time before symptoms develop. And can we slow the disease process down? And what's happening in that area. So what is dementia? Well, we use the term to describe somebody who's having difficulty with their usual activities due to problems with thinking, memory, understanding, speaking and organising, etc. They have problems with community activities, might be their job, might be their social activities, it might be shopping, cooking, handling money, finding their way around, making themselves understood and understanding the situation. And eventually dressing and bathing and personal care. What about Alzheimer's disease? Well, this is Alzheimer. He was a psychiatrist and pathologist who was working in Germany 120 years ago, a time of great progress in understanding what the brain looked like under the microscope. And in 1906, he described the brain changes seen under the microscope in a patient with dementia and described the disease that later bore his name. Now, what did he find? He found a combination of two features. On the left, you can see a sort of fried egg arrangement here, and that's called a neuritic plaque. And then on the right here, you can see a sort of tadpole-looking thing, and that is a dense collection of protein inside a nerve cell, and that's called a neurofibrillary tangle. And this combination of plaques and tangles in the brain of a person with dementia is what we, strictly speaking, what we mean by the term Alzheimer's disease. Now, if you looked at the brains of 100 people with dementia, between 50 and about 70 of them would have plaques and tangles. Now, some of them would not have plaques and tangles, they have another disease process, and some of them would have other disease processes as well as plaques and tangles, but about 50 to 70 of them would have plaques and tangles. So that's why sometimes you will hear the terms Alzheimer's disease and dementia used interchangeably. Now, fast forward about 70 or 80 years, and the chemical structure of these proteins was worked out, and the plaque was found to be a protein called beta amyloid, and the tangle was made of a protein called tau. And interestingly, a lot of the early work on beta amyloid was done by an Australian, Professor Colin Masters, in Germany and the UK, Germany and the USA, and he's still working on amyloid here in Melbourne. Now, where do these proteins come from? Well, our proteins are made by genes. The genes are a recipe for all the proteins in our body. Now, for some decades, it's been noted that sometimes Alzheimer's disease and other forms of dementia run in families. And this is one such family. And you can see that the black symbols indicate the disease being transmitted from one generation to the next. Not everybody in each generation is affected, but there is a transmission from each generation to the next. You can see these are the ages at death and the ages at onset, and you can see that the people who are affected haven't survived as long as 60, and the people who are affected in middle age, commonly between their 30s and their 50s. So it's quite a young, usually quite a young onset disease. When we see that pattern, it makes us think that the cause of this inheritance pattern is a problem with a single gene. And after a long search, in 1991, the first gene was found in a family with Alzheimer's disease from Britain. Since that time, there have been several hundred families around the world identified with a genetic cause for their Alzheimer's disease. And we know of about 30 families in Australia with one of these genes. Now, why do these rare families hold the key to understanding the biology? Well, unlike most people with dementia or Alzheimer's disease, they have a single known cause, which is a faulty copy of a single gene. So studying the gene in the laboratory and what it does should give us a handle on what's happening in the brain in people as they develop symptoms. Now, in 2008, an international study was set up led by Professor John Morris from Washington University in St. Louis. There he is there in the white coat, with funding from the US National Institutes on Aging. And they started with some centres in America, in London, and 3 centres in Australia, which Sydney, Melbourne and Perth. The Sydney site is here at NeuRA and is headed by Professor Peter Schofield. Now, the idea was to find a marker of disease activity that would tell us in these families when the disease process became active. Now, after a short time, a couple of years, the initial results were published in the New England Journal of Medicine. And this is what I'd like to share with you because it's the most significant finding. And this is a type of brain scan which measures the deposition of amyloid in the brain. And you can see in the top right-hand corner here, this is somebody who is at the age where they're developing symptoms. And you can see that there's a lot of colour in the brain compared with somebody of the same age who is not developing symptoms. And on the left here, we have, in the top row, we have somebody who is from one of these families, 20 years younger than their parent was when they developed symptoms. And this person is carrying one of the genes, and this person is a non-carrier. There's not very much difference between those two scans, but you can see, I think, a little bit of yellow there. When we get to 10 years before the symptoms develop, you can see that there's quite a bit of yellow and green and not very much purple in the person with the gene compared with a person of the same age without the gene. And then this is at the very time, these people here are functioning perfectly normal memory, no problems with their daily activities, but they've got amyloid in the brain. So we can see from these pictures that the amyloid deposition, amyloid gets deposited over a long period, 15 to 20 years, before people get any symptoms. So what if we could do something between B and C or between A and B? In the 10 to 15 years before symptoms, what if we could do something to prevent the symptoms happening? Well, one thing we could do is to try and stop the amyloid accumulating or removing the amyloid that's there. And this was the next step in the Diane study. They've set up a trials unit with funding from the US Alzheimer's Association. They've got some pharmaceutical companies to try who each have their drug that they want to try in this group. And with the cooperation of the families concerned, this has been going now for several years. And these are the sites of Diane and Diane 2 that are currently in existence. And in blue, you can see that there are more sites joining up as we go along. Now, how can we get rid of amyloid? Now, if you can think of a very simple picture, supposing the brain was a sink and it was full of amyloid. Well, one thing you could do would be to pull out the plug and enlarge the plug hole and get rid of it that way. Another thing you might do would be to turn off the tap. Now, drug companies have worked out potential drugs that could do either of those things. But as it happens, the DIAN-2 has started with 2 drugs that remove amyloid by sort of enlarging the plug hole. And about 140 people carrying their family mutation have been enrolled worldwide, and they are taking either one of these drugs or an inactive placebo compound. And the analysis of that trial will begin when everybody in the trial has done at least 4 years of treatment. So it's not a 6-week trial. The people are in it for the long haul. And the analysis will begin at the end of 2019. What evidence do we have that this might work? Well, this is from another study and from a similar drug which has been used in people with the early stages of Alzheimer's disease. And this was a preliminary study published a couple of years ago. You can see on the left people at the beginning of the trial, and they all have amyloid in the brain. After 1 year of treatment, you can see that the placebo group here has had no change in amyloid, whereas people taking increasing doses of the experimental compound have had substantial clearing of amyloid. So that's one bit of good news. Does it cause symptoms? Does it cause symptoms to improve? Well, this trial wasn't quite big enough to show that effect. However, there was encouraging evidence that people on the active drug, particularly on the higher doses, did not decline in the same way that the people on the placebo did. So there is encouraging evidence, but it needs to be confirmed by larger studies. We are hoping that DIAN-2, because it is a prevention trial and starts earlier, will have a better chance of success. My thanks go to the families who have endured so much and who have contributed so much to research over the last 30 years. People like Amanda turn up cheerfully every year for their extensive battery of tests, scans, blood tests, and even a lumbar puncture for spinal fluid. 30 years ago, there wasn't very much hope for people in these families, but now, with the arrival of these prevention trials, I can see that there is hope developing, and that the younger generation particularly has something to look forward to. So our task is to keep going and to find a means of prevention, if not for the current generation, then for the next. And thank you very much for listening.